The great fungi Phenohunt. I mean, why are we going full mushroom matchmaker? Why spend months or even years hunting, isolating, and breeding fungi? Can't we just buy a spore syringe or liquid culture and call it a day? Well, you could, but where's the fun in that? More importantly, where's the genetic diversity? If we want to create something new, resilient, or just plain weird, we're going to need to play with nature's building blocks. Here's the goal. Find more wild fungi breeds because wild strains have more unique traits. We'll have to germinate their spores by either cloning and collecting spores from a fruiting body or collecting spores from the wild specimen. Once we do this, we need to isolate those Monokaryons, which is just a fancy word for half genome, or one parent's genetic information. Once we confirm under the microscope that the germinated mycelium we have is a monokaryon, we can breed two genetically different monokaryons to form a dikaryon, which would be a mated cell. This full cell can actually produce fruiting bodies, and that's what we want to get to combine the two parents and get new traits. This hybrid will just be a combination and random selection of these genetics from both parents. It's like Tinder, but for mushrooms, and we're gonna need a lot of agar. Let's go hunt for some more wild species. And we're out trying to find some more wild oysters for this Phenohunt project, our custom breeding project. I need to find some more genetic diversity. So as we start to break out into season here, we're just keeping an eye out on all of our local trails, trying to find some nice pleurotus or some oysters that we can collect some wild samples and take home. But I have not been able to find any yet, so I have this other place in mind, so we'll have to hit there next and see if we can't find more. We also found while we were out fossilized a shell of some sort. You can see what it is. And you can see these cool little bits right there, this texture right here. I'm not really sure what that is. Super neat though. I've been hiking for miles and still nothing. Look at this though. I think I found something. Look at these beautifully found lion's mane and blue oysters. I wonder where they came from. This is such a strange occurrence. Just this flawless cluster on this incorrect tree with no mycelium. It's so strange how nature works. Must have been the rain that brought them here. This is why science requires peer review. All jokes aside, however, these are just some store-bought oysters and lion's mane, blue oysters and lion's mane, which you could do for your own Phenohunt. I've yet to forage my own wild species, and realistically all we need is just a different genetic source from what we have currently. So yes, even these store-bought mushrooms are still genetically diverse, and we can use them to start the Phenohunt. Let's go ahead and collect this forage and get them back to the lab. We're just we have our two wild samples here, our blue oyster and our lion's mane. We're gonna grab some aluminum foil and go ahead and take a nice spore print of both of them. And we're gonna do this in front of the flow hood so they hopefully dry out a little as they do this as well. We just have a little bit, we picked one of the more mature samples from both. We're just gonna set them spore side down. I might use like this wire to raise them off the surface just a tad, you know. Perfect. So now we'll just go ahead and let those set in front of the flow hood for a while and collect some of those spores. After, we'll go ahead and do some traditional swabbing. I have not been able to find bamboo swabs yet, so I'm going to try sealing just some regular paper swabs and see if they hold up after sterilization. I'm also going to try something that I'm going to call the mega swab. This may already be a common technique, but I have these cotton balls, which I think I can just take one, I can sterilize them, take one, swab a plate down, and then use tweezers, those ultrafine tweezers from before, and do that streaking method. So let's go ahead and get these spores. Now to prepare the swabs and the balls, it'll actually be super easy. We're just gonna grab a handful of these. We got a handful of swabs already in there. And we're going to harvest one of these old metal lids and take one of these filter patches. There we go. Cover this other side and pull this through. Perfect. We'll go ahead and throw these in the sterilizer probably for like 45 minutes. I can't imagine they're going to be very dirty or that'll need to go very long. We're using this lid just because there is a filter patch on it though. Cover the lid in aluminum foil and send it off in the pressure cooker. And not that it probably matters too much, but I'm going to go ahead and set these over top of each of these overnight just to kind of let them build humidity, just in case that helps. And I'll remove them tomorrow and let them dry out a little bit longer. Now, with our spores, we need to check the viability. So we have our blue oyster and our king oyster here, and 2 sterilized, individually packaged swabs. Use some sterile tweezers to rip off a piece and swab these into our water agar plates, which we have had cooling down in front of the flow hood right here. So let's go ahead and get that started. Start with this king oyster. Got a clean plate set off here to the side, and we're going to pop open our syringe. This is a nasal syringe, so it's not really exactly what we want. Hopefully there's spores on that. With our sterilized tweezers, we'll go ahead and grab a little bit off the end of this. Now this is not your traditional cotton swab, so it's not going to be exactly the same thing, but hopefully it should do the job just the same. We'll grab just the smallest amount that we're able to in the end of these tweezers, grab our plate, and we're going ahead and do an S pattern throughout the agar, kind of trenching this in there, and at the very end of the S, we'll release and leave that piece of swab in there. So we're going to start up here at the top. And that was messy, but for a first attempt it shouldn't be terrible. Hopefully there we'll get some dikaryons and some monokaryons towards the end or the middle, and those will be the isolated spores that have germinated, but it is just one parent, so it's only one spore and not a dikaryon cell, so we can isolate that and breed it with another monokaryon to have 2 genetic parents. We'll go ahead and label this, flip it upside down, seal it up, and let this incubate for a few days. And we're going to repeat this one more time. Just trench, grab and drag, and release at the end. Oh, I don't have too high expectations for these plates. So in the meantime, I am going to go pick up some proper swabs that I can sterilize and use them correctly. But we'll go ahead and see how those germinate over the next few days. With all 3 of these plates all hopefully successfully grab and dragged, I'm going to throw them into incubation. We are going to run to the store and we're going to pick up some proper cotton swabs with bamboo sticks so we can run them through the sterilizer and have a little bit more of a proper swab to use. I just swabbed these blue oysters and labeled the swab packet. We are going to repeat the same process, grab and drag through a few of these plates. And here is our 3 blue oysters done. And we'll go ahead and set those to incubation with the other ones. So we have the sterilized cotton balls and swabs. They all held up really well. We're going to go ahead and remove, smells like, very much like mushrooms, the lid from both of these. A lot of lion's mane spores. Don't really see oyster spores, but we may just pull a direct swab off of that as well. Alright, we have those spore prints hopefully drying out a little bit. I don't really see too much on the blue oyster, which isn't a big deal. I will leave it on there for a little bit longer, but I'm also going to grab a swab from it. The lion's mane already has this really nice white print, I can see it, but I don't see anything from the blue oyster, which is not a big deal, but we have that sterilized jar. We're just going to do a normal swab since these held up alright. Now with this, we'll just grab our sample, and we're just going to roll it through the gills, and we'll let that dry out here for a little bit. I'm honestly probably going to do both sides of this swab. We'll let that dry out. While we are right here, We'll grab one more, and we'll go ahead and get a nice swab of the print that's already here on the ground for this lion's mane. And again, I'm going to do both sides of this Q-tip and let that dry out as well. And we can also take a peek at some of the first swabs I did, which I'm not really too confident in these because of the material of that swab and probably the quality of the prints. It's only been a few days, so we'll give them a while to see if they germinate anything, but I'm not too hopeful, so we'll go ahead and do some more streaking with these new swabs. Now with these spore prints hopefully completed, I'm not sure if you can see, there's a lot of white fuzz. I'm going to actually save these specimens and let them go a little bit longer. Now I've let these go a little bit longer and the swabs have dried out, so I'm going to remove the swabs and give them an area. The lion's mane in the front, we have the blue oyster in the back, and I'm going to remove both of these specimens to close and label these. We can go ahead and put these into storage for now, and these specimens can just be thrown out for now, and then I'll re-sterilize my hands. With the hands re-sterilized, we're going to go ahead and grab our 2 plates for these swabs. These are the same as earlier, just water agar. It's like 4 or 5 grams of agar, I think, to 400 or 500 milliliters of water. Start by removing all this tape for this first one. Take our tweezers. We're going to go ahead and sterilize those in the induction sterilizer. You can use a flame sterilizer or whatever other means you have of ensuring there are no contamination on your tweezer tips. Let this cool down for a second. We're just going to grab a singular strand off of these. This front one is the lion's mane. This back one is the blue oyster. I think I'm just going to go ahead and grab a Ziploc bag to store those in afterwards. Actually, I lied. I had these little centrifuge tubes, which I'll use to go ahead and throw the swabs in so I can label them and just store them for now. Could be a pretty useful method. I'll need 2 of them for now. We sterilize our hands real quick. Sanitize isopropyl alcohol. Now we'll take our lion's mane swab. We're going to grab just the tiniest singular thread off the tip. It's definitely too much right there, but it'll do. I don't really care for the longevity of these. We'll pop open this plate, reposition our swab, grab and release. Make sure that little piece stays in here. Getting a little messy, but that should work. We'll sterilize our tweezers again, and while those cool down, we'll throw this lion's mane swab into its tube. Go ahead and label this. This labeling is super important. It lets me backlog my work, especially when I'm doing years worth of work. I can go back, find my isolations, where I got them from, and it makes the whole breeding process just a little bit easier for me, and have a nice backlog of what I've done. With this cooled down, let's go ahead and grab a tiny strand off this blue oyster, and we are going to reposition the plate a little bit. Drag it through. And at the very end, release that piece in there and close it back up. Go ahead and cling wrap this. And cling wrap the lion's mane. And we'll wait for germination. Along this line, we're definitely going to see a mix of both Monokaryons and Dikaryons. Towards the middle and the end is where we're going to hunt for hopefully some nice Monokaryon cultures that we can transfer from here onto a new plate, and then observe under a microscope to confirm that there is no clamps. But for the meantime, let's go ahead and move these off to incubation. I'm a lot more confident about these new plates with these paper swabs that end up working out in a jar, which I now have a sterilized jar of swabs and balls. And we'll move this blue oyster swab into one of these tubes just for safekeeping. We'll move that into the culture box. Now, I think at this point we may have a few putative monocultures. I haven't confirmed them under a microscope yet, so what we need to do is transfer just a small sector from the leading edge of those little cultures from the grab and drag plate move those to new water agar plates, and wait for germination there. My newer plates have yet to see any germination. I think that this is just because I've been using water agar as opposed to any kind of nutritional. So this agar is just about 4 or 5 grams of agar powder into about 500 or 400 milliliters of water, I don't remember which, and there's no nutritional supplementation in there. So it's taken a while for the spores to germinate, but it's important that we let them grow out, get them under a microscope so we can ensure that there's no clamp connections, which means that they are a monokaryotic cell, and then we can breed them down the line. It's going to take a while for us to isolate a few of these Monokaryons though, so it's important that we try to get as many cultures done as we can, and isolate as many monos as we can as well. Let's go ahead and transfer the leading edge off of all of these putative monos to some new plates, and let them grow out. Now, sorry for the flashbang, and this might be hard to show you, but you'll see towards the end of our grab and drag, there is a few spots along this line where there's been some germination. Hopefully there's going to be some monokaryotic cultures that we can transfer to some new plates and confirm them under a microscope once they've grown out. Now we have our 2 king oysters here, which both have putative monos, what I think are monos. I'm not going to view them under the microscope yet because I want to isolate them and grow them out a little bit further first. So we're going to sterilize our scalpel here, let it cool down. Right here we have all of our new water agar plates and our old grab and drag plates right here. And it's going to be hard to show you. There are a few spots of germination on both of these king oyster plates. I don't really think you're going to be able to see them because of the condensation, but we're going to go ahead and unwrap them and get them transferred onto some new plates. And we're going to start here with this king oyster. Go ahead and start by unwrapping it. And now we will bring our new plate up front closer to the flow hood. Take our nice clean scalpel blade, and we're going to take, smaller than the tip of the pen, like the smallest amount that we can grab on the tip of this, on the leading edge of one of these germination spots. Just the leading edge of one of those, and put it into a sector over there. What I'm actually going to do is use tweezers. I think this will be a little bit easier for me to grab just that leading edge. So I will sterilize these real quick, let those cool down. And now with the tweezers cooled down, we'll come in to our plate, look for another germination spot, just the tiniest bit, and do this into another sector over here. Sterilize the tweezers again, and I see one more at the end that I'm going to target now. Pop this off. Grab just a little bit from the leading culture. Sector that one off. Sterilize. We want to make sure that we're sterilizing our tool between every single grab, because if there is a chance that we get one cell that touches another monokaryotic cell, it's no longer monokaryotic. Those cells are going to mate, they're going to transfer genetic information, and we're going to be left with a dikaryon cell. I want to see, I've got 3 sectors off of this. I want to see if I can get one more to get onto this plate. So let's observe this plate again. I grabbed and isolated all the cultures that I can see off of that plate. And the ones on this plate, there is mycelium, but it's slightly off, and I'm just worried about it being some kind of contamination, so I'm not even going to mess with it or worry about trying to grab that. This one will go into the trash, and we're going to go ahead and let this one go to the trash as well, just because it's the one that we already grabbed and pulled our putative monos to this secondary plate. That does mean I do need to print off a new label. Alright, and we have the new label printed for our putative mono plate. We need to let those grow out for a little bit before we isolate them and then confirm under a microscope. Now this can be wrapped, and we'll put this plate back in incubation for a few days to see how those sectored off bits. I don't know really how well you're going to be able to see them. There's 3 or 4 sectors I have in there, and see how those expand. I'm also going to do one of these mega swabs with this older lion's mane print that I have. I'm not really sure the viability of it or anything. This is from December of last year. But I'll show you what I was kind of talking about with these bigger swabs if you don't have Q-tips. Ultimately, though, I think the Q-tips just make things a lot easier. So, not really a significant reason to be doing this technique, in my opinion. And we are going to go ahead and take just a bit. I mean, you could either probably swab this directly. And now for this mega swab, we're just going to take a small bunch, literally just a few strands, and get that on there. And we're going to swab over this print. And we're just lightly dragging the cotton threads through the spore print. Hopefully we got some spores on there. And what we'll go ahead and do is use a new plate and do the same grab and drag. So close this, move out of my way for now. And we are going to take this thread with hopefully some spores onto it and complete just another simple grab and drag and release. And maybe a better way to use these, which I don't care about saving this print, so I'm going to try this another way, which is just I'm going to open up the inside of this and I'm just going to swab down this entire print, and I'm going to come through, grab just a little bit off of there, and we can swab this into another plate as well. I'm not really sure why you'd need to do the grab and drag with a big cotton swab like that, but if that's all you have access to, it should work just the same. We're going to come through and gently swab through here, releasing the piece of cotton at the end and closing it all up. Go ahead and sterilize it all and let that cool down while we get these first 2 labeled unwrapped. And this can all be tossed. You could save the swab if you wanted to, but again, I'm not really confident in the viability of these spores. They're a little bit older, and I don't believe that I would have collected them properly anyway. I didn't have the flow hood then, so I don't think that these spores dried out properly before they were stored. You could keep that swab though, but I have enough other prints to work with and enough other growing right now that I'm fine with throwing that away. Let's throw these labels onto these lion's mane swabs, these lion's mane grab and drags that we did, and let's get those into incubation and we can monitor them over the next few days. And like I was explaining with those previous swabs, I'm not entirely sure that the spores are in great condition, so since we did not check the viability first, we went straight to grab and dragging, we're going to have to wait to see if they germinate. And then after we isolate the putative monos, let those grow out, observe them under a microscope to ensure that we are actually dealing with mushroom mycelium, and also confirming that it is a monokaryon culture. This video is just the start to this project and will probably end up being years worth of work throughout different kinds of species and isolations that I work on. One of my goals is to find local species that I can harvest, clean up, and isolate, both to breed with and to isolate the original for preservation purposes. Once we have a few monokaryon cultures of the same species isolated, we can go ahead and start crossbreeding them to dikaryons, and once we fruit some dikaryyon cultures, we can pick the fruits that we like, collect spores, take tissue cultures, and advance our Phenohunt. After a few generations of fruiting, we'll be able to use different crossing techniques to reinforce the physical characteristics that we like, and hopefully reinforce those traits to get ourselves a nice, reliable, strong, clean culture. Now this is a little bit of a longer video, but what I'm going to be working on over these upcoming weeks is to continue to grab and drag different spores, isolating these monokaryons, and in the next video we're going to crossbreed these monokaryons and observe them under a microscope. Until then though, thank you all very much for joining, and we'll see you in the next one.